Hi dear students, today I am going to talk about Calvin cycle of photosynthesis, the amazing process of carbohydrate synthesis in green plants. What is Calvin cycle? Calvin cycle is the fundamental cycle in dark reaction only through which glucose is synthesized from carbon dioxide. That is, organic carbon of glucose is synthesized from inorganic carbon of carbon dioxide using photochemically synthesized ATP and NADPH. This cycle was discovered by Melvin Calvin, an American biochemist. That is why the name Calvin cycle. He discovered the cycle with the help of radioactive carbon isotope C14 and conducted studies in green algae, chlorella and Cnidesmus and elucidated the complicated cyclic pathway of carbon reduction. For this amazing work, he was awarded Nobel Prize for Chemistry in 1961. This cycle is also known as C3 cycle since the first stable compound formed here is a 3-carbon compound. The cycle is also known as photosynthetic carbon reduction cycle or C3-PCR cycle or path of carbon in photosynthesis. So let's see the different steps in Calvin cycle. Calvin cycle is a cyclic enzymatic process completed in 4 steps. The first step is carboxylation of carbon dioxide acceptor compound or fixation of carbon dioxide. Here, a 5-carbon compound known as RUBP, ribulose 1,5-bisphosphate. It acts as the carbon dioxide acceptor in Calvin cycle. The second step is reduction. The third step is glycolytic reversal and formation of hexose sugar. The formation of hexose sugar takes place by the reversal process of glycolysis. That is why the term glycolytic reversal and formation of hexose sugar. The fourth step is the most important step, regeneration of carbon dioxide acceptor, that is the RUBP, which is essential for maintaining the sustainability of Calvin cycle. Since the carbon dioxide acceptor reserve in stroma is limited, regeneration is highly significant for the continuous operation of C3 cycle. So these are the 4 steps in Calvin cycle. Carboxylation of carbon dioxide acceptor, second step is reduction, third step is glycolytic reversal and formation of hexose sugar, the fourth step is regeneration of carbon dioxide acceptor. So now we are moving on to the reaction part of C3 cycle. Here, first phase is carboxylation. Here, carbon dioxide, it is accepted by the carbon dioxide acceptor compound, that is RUBP. Here, 6 molecules of RUBP are utilized for the carboxylation process. Here 6 molecules of carbon dioxide are coming and they are accepted by 6 molecules of RUBP. And here RUBP is nothing but a 5-carbon sugar, that is a pentose sugar with 2 phosphate groups. Bisphosphate means 2 phosphate groups on both sides. So ribulose bisphosphate, RUBP is a pentose sugar with 2 phosphate groups on both sides. It is a 5-carbon compound. So 6 molecules of 5-carbon compounds are there. These 6 molecules of RUBP accepts 6 molecules of carbon dioxide. So 36 carbons, 30 carbons plus 6 carbons. So 36 carbon are coming. Here they are first forming, initially they are forming a 6-carbon addition compound which is quite unstable and soon dissociated into 12 molecules of 3-phosphoglyceric acid. 12 molecules of 3-phosphoglyceric acid. This is the first stable compound of Calvin cycle. 3-PGA, 3-phosphoglyceric acid. This 12 is the number of molecule. So 12 molecules of 3-phosphoglyceric acid are forming. This is because one molecule of RUBP combines with one molecule of carbon dioxide, then it formed one molecule of 6-carbon addition compound. When a 6-carbon addition compound break into 2 molecules of 3-phosphoglyceric acid. Like that, 6 molecules of 6-carbon addition compound, when breaking up, it form 12 molecules of 3-PGA. And this carboxylation process, it is catalyzed by an amazing enzyme called Rubisco. I will explain more about Rubisco later in this presentation. So this step is represented as the carboxylation step. RUBP combines with carbon dioxide and initially a 6-carbon addition compound is formed which is unstable, which break up or dissociates into 3-phosphoglyceric acid. It is a 3-carbon compound, but the carbon number is same. Here 6 into 6, 36. Here also 12 into 3, 36 carbons. Okay. Now the product of carboxylation, that is the first stable compound of C3 cycle, that is the 3-PGA. So this 3-phosphoglyceric acid, it is now undergoing reduction. So next phase is the reduction phase. Reduction phase is also very important. All of us know the dark reaction itself is a reduction phase in which carbon is getting reduced to carbohydrates. So the actual reduction is happening in this phase. So here the reduction is completed in 2 steps. The first step is actually an activation process in which this 3-phosphoglyceric acid is converted to 1,3-bisphosphoglyceric acid. That means one more phosphate group is added to this 3-PGA. So this extra phosphate is getting from ATP. That is why along with this phosphate group, some energy is also provided to this molecule. So this reaction is happening along with the ATP hydrolysis. So 12 molecules of ATP are hydrolyzed to release 12 phosphate groups, inorganic phosphate. They are added to this position. That is why 1,3-bisphosphoglyceric acid is forming and 12 ADP is regenerating. Then the 1,3-bisphosphoglyceric acid now formed, it is undergoing the reduction process, the actual reduction process. Here the reduction means reduction is the addition of hydrogen. Here the hydrogen is supplied by the NADPH produced in light reaction. So for the reduction of 1,3-bisphosphoglyceric acid, 12 molecules of 1,3-bisphosphoglyceric acid, 12 molecules of NADPH are utilizing. So it provides hydrogen to this reaction. So the reduction is happening. So when reduction takes place, what happens? Here glyceric acid, here the functional group is acid, COOH or COO minus. When an acid undergoes reduction, it will form aldehyde. This is the rule in organic chemistry. Aldehyde is formed as a result of reduction to acid group. So when glyceric acid undergoes reduction, it will form glyceraldehyde. So this is the rule in organic chemistry. So as a result of reduction, glyceraldehyde is forming. From glyceric acid, glyceraldehyde is forming. And one more thing happens. Here the extra added phosphate group is now removed from here, because its function is just activating the molecule. So after this function, the phosphate group is removed from here, and also water molecule is also forming as a result of this reaction. So both of these components are removed from this reaction. So the remaining product is 3-phosphoglyceraldehyde, or it is also known as glyceraldehyde 3-phosphate. 3-phosphoglyceraldehyde or PGAL, or it is also represented as glyceraldehyde 3-phosphate or G3P. Both are same. Okay. Here when reduction takes place and the glyceraldehyde is formed in stroma, actually the first sugar molecule is synthesized here, because it is the simplest form of sugar. Glyceraldehyde is the simplest form of sugar. It itself is a monosaccharide. It is a triose sugar. So the simplest form of monosaccharide glyceraldehyde is now formed as a result of this reduction process. Okay, so this reduction process, which is occurring in two steps, it requires the presence of two enzymes also. The first step, that is the phosphorylation aspect, it is catalyzed by the phosphoglycerokinase enzyme, and the second part, the dehydrogenation, the reduction process, is catalyzed by triose phosphate dehydrogenase. From this 12 molecules of 3-PGAL or glyceraldehyde 3-phosphate, we have to synthesize glucose or fructose, that means the hexose sugar, and also we have to regenerate the RUBP. So out of these 12 molecules, 2 molecules of 3-PGAL or glyceraldehyde 3-phosphate, I am repeating, PGAL and glyceraldehyde 3-phosphate, both these terms are similar. They are representing the same compound. Okay, so here 2 molecules of 3-PGAL are taken for the synthesis of hexose sugar, and this synthesis of hexose sugar from 2 molecules of 3-PGAL, it is happening through the reversal process of glycolysis. That is why this process is known as glycolytic reversal and formation of hexose sugar. Actually, what is happening is that one molecule of PGAL out of this, it is forming the isomeric form of PGAL, that is DHAP, dihydroxyacetone phosphate. So this PGAL and DHAP combine to form fructose 1,6-bisphosphate first, then it is converted to fructose 6-phosphate, which is then converted to glucose 6-phosphate and then to glucose. So this is the reversal process of glycolysis of cellular respiration. So that is why this process is termed as glycolytic reversal and formation of hexose sugar. So through this process, hexose sugar is formed in C3 cycle. Okay, now the remaining 10 molecules of 3-PGAL or glyceraldehyde 3-phosphate are going to be utilized for the synthesis of or for the regeneration of RUBP. This is very important for the continuous operation of C3 cycle. So this regeneration happens through a complex chain of reaction. So this is not simple as like this. This is a very complicated process which involve several intermediate compounds such as erythrose phosphate, cellulose phosphate, ribose phosphate, pseudoheptulose phosphate, ribulose phosphate, like that. So several intermediate compounds are there, several combinations and recombinations are taking place here, and as a result of this complicated process, at last 6 molecules of RUBP are regenerated. So in this last step of this regeneration, it requires 6 molecules of ATP to be hydrolyzed here. So here a total of 18 ATP are needed, 12 ATP for this reduction process and 6 ATP for this regeneration process. So the entire C3 cycle pathway requires 6 carbon dioxide, then 12 plus 6, 18 ATP and 12 NADPH for the synthesis of one molecule of glucose. And at the same time, per carbon, when we think about carbon, so in order to fix one carbon dioxide through Calvin cycle, how many ATPs and NADPH are needed? For fixing one carbon, for fixing 6 carbon, 18 ATP plus 12 NADPH. So for fixing one carbon, we require 3 ATPs and 2 NADPH. Okay. So here ADP and NADP plus are regenerating in this cycle. They are utilized in the light reaction process again for the synthesis of ATP and NADPH. So that's all about this reaction part of Calvin cycle. Let's see another representation of Calvin cycle. Here we can see the stepwise processes. So just revise what we learned. The first process is carbon fixation, as we learned. This is the carboxylation process where carbon dioxide binds with RUBP with the help of Rubisco. So a 6 carbon molecule is first formed, initially formed, which is unstable, which splits into 2,3 carbon compounds. This is the first stable compound of C3 cycle, that is 3-phosphoglyceric acid. So you have to remember the first stable compound 3-PGA. So next phase is the reduction process through which 3-PGA is converted to glyceraldehyde 3-phosphate or 3-PGAL. So this is a two-step process in which ATP and NADPH are utilizing. So as a result of this two-step reduction phase, glyceraldehyde 3-phosphate or PGAL are forming. Here 12 molecules of 3-PGAL are formed, and out of this PGAL reserve or G3P reserve, 2 molecules are taken for carbohydrate formation through the glycolytic reversal process, and the remaining G3P or remaining PGAL are utilized for the regeneration of RUBP, and here ATP are needed for this last step of regeneration. Okay. Let's see another representation which will help you to understand the carbon balance sheet of C3 cycle. So while studying dark reaction cycles, you have to understand correctly the carbon balance sheet. So here we are starting from RUBP. RUBP is a 5 carbon compound, and we are taking 6 molecules of RUBP, means 6 into 5, 30 carbons. When it combines with 6 carbon dioxide, 6 into 1, 6 carbon. So 30 plus 6, 36 carbon are now present. So when 12 molecules of 3-PGA are forming, 12 into 3, here again 36. Then when it forms 12 molecules of 3-PGAL, here also 12 into 3, 36. So out of this 36 carbon, 2 molecules of 3-PGAL are taking for the synthesis of glucose. So 2 molecules of G3P means 2 into 3, 6 carbon. So the 6 carbon is now here in the form of a hexose sugar. So the remaining number of carbon is 30, because 6 carbon were taken for the synthesis of glucose. So remaining 30 carbons, that means 10 into 3. 10 molecules of 3 carbon compound. So 30 carbon are now present here. They are utilized for the regeneration of RUBP. Here from this 10 molecules of 3 carbon compound, we have to synthesize 6 molecules of 5 carbon compound. So 6 into 5, 30. So number of carbon is same, but for this production or for this regeneration of 6 molecules of 5 carbon compound, so many combinations and recombinations are needed. Several intermediate compounds are required. So through this complicated process, the 6 molecules of 5 carbon compound are regenerated. So this is the carbon balance sheet of Calvin cycle. So it's all about C3 cycle. Now we have to learn a little more about the wonderful enzyme Rubisco. This is the structure of Rubisco. This is the enzyme responsible for carboxylation process of Calvin cycle, and it is an enzyme. Actually, the actual name of enzyme is ribulose 1,5-bisphosphate carboxylase bar oxygenase. So it is actually, Rubisco is an acronym for this ribulose 1,5-bisphosphate carboxylase bar oxygenase. Acronym is nothing but abbreviation which is spelled like a single word. So here, like all other enzyme names, here also the substrate, the substrate for this enzyme is nothing but RUBP. So this is the name of the substrate, that is ribulose 1,5-bisphosphate. The second part is the reaction which is catalyzing, that is carboxylase bar oxygenase. This is because of an unusual property of Rubisco, because this enzyme possesses a dual property. It can catalyze both carboxylation and oxygenation of RUBP. That is why this name. Rubisco is the acronym for ribulose 1,5-bisphosphate carboxylase bar oxygenase. So you have to remember this name. Then Rubisco is the most abundant enzyme or plant protein on earth. This is very important. It is the most abundant enzyme on Earth. And it fixes 200 billion tons of carbon dioxide annually. This Rubisco is responsible for fixing 200 billion tons of carbon dioxide annually. And it accounts for 40% of the total soluble proteins of leaves. This is also another important feature of Rubisco. And about the structure of Rubisco. Rubisco is actually a complicated or it possesses a complex structure with 8 large subunits and 8 small subunits. The large subunits are encoded by chloroplast genome and they are synthesized in ribosomes present in stroma or the chloroplast ribosomes. And these large subunits are actually possessing the active sites of Rubisco. Then the small subunits of Rubisco are encoded by the nuclear genome and they are synthesized in the cytoplasmic ribosomes and later assembled inside the stroma. So this is about the wonderful enzyme Rubisco. This picture explains the unusual property of or the dual function of Rubisco. Rubisco possesses the property of catalyzing carboxylation and oxygenation of RUBP in some environmental situations. Carboxylation, when Rubisco catalyzes the carboxylation of RUBP, the normal situation, that is the carbon dioxide is added to the active site, and as a result of the normal carboxylation 3-phosphoglyceric acid are forming. So the normal C3 cycle is operating here, no differences. But when oxygenation takes place, oxygenation of RUBP is taking place in some unusual situations like high temperature, high oxygen concentration, high light intensity, etc. In such situations, Rubisco catalyzes, Rubisco favor oxygenation of RUBP rather than carboxylation. So in such situations, oxygen molecule are occupied in the active site of Rubisco. So oxygenation takes place, and when oxygenation of RUBP is taking place along with 3-PGA, another molecule is forming, that is 2-phosphoglycolate. This molecule acts as the starting compound for an unwanted cycle, that is C2 cycle of photorespiration, which is added to C3 cycle and it is causing many problems. Rubisco is an very heavy protein with 560 kilodalton molecular weight also. So these are all about the unusual properties of Rubisco. So this is a very important enzyme regarding C3 cycle. So you have to remember all the points regarding Rubisco. Okay. So what are the significances of C3 cycle? This amazing process, this amazing cyclic process taking place in the stroma, it takes carbon dioxide molecules directly from air and convert them into organic matter called sugars. So this process can be considered as nature's most sustainable production process also. And all photosynthetic eukaryotes, from most primitive alga to the most advanced angiosperm, reduce carbon dioxide to carbohydrate through this basic mechanism. And all other photosynthetic pathways, C4, CAM and C2 pathways, are either auxiliary or dependent on C3 cycle. So the C3 cycle is very important regarding the ecosystem or biosphere and for the existence of all living creatures on Earth. I hope everybody understood the process well. So shall we revise the important points? Who discovered C3 cycle? Melvin Calvin. The initial carbon dioxide acceptor in C3 cycle, it is RUBP. Name the enzyme involved in carboxylation of C3 cycle. It's Rubisco. The first stable compound in Calvin cycle, that is 3-phosphoglyceric acid or 3-PGA. How many ATP and NADPH molecules are required to synthesize one molecule of glucose in C3 cycle? 18 ATP and 12 NADPH. How many ATP and NADPH molecules are required to fix one molecule of carbon in C3 cycle? It is 3 ATP and 2 NADPH. So we learned about a wonderful process taking place in plants that provides energy in our food, which powers the entire biosphere. So I hope all of you enjoyed. So thank you all.